Herzlich willkommen zur heutigen Veranstaltung, dem Einführungsvortrag eines neuen Kollegen im Departement Umweltsystemwissenschaften, kurz D-USYS genannt. Mein Name ist Nina Buchmann und ich bin Vorsteherin des D-USYS. Dieses Frühlingssemester ist sehr ereignisreich und wird ein sehr ereignisreiches Semester werden, vor allen Dingen im Hinblick auf diese Einführungsvorlesungen von neuen Kollegen und Kolleginnen. Das D-USYS wird dieses Semester alleine im Frühlingssemester 4 neue Kollegen und Kolleginnen begrüßen dürfen. Das dürfte wahrscheinlich Rekord sein für unser Departement. Und heute darf ich Professor Dr. Thomas Ward Crowther ankündigen, der dieses Rekordsemester einleiten wird. This semester is probably record holding for USYS because 4 new colleagues will be introduced and Tom is the first one. Tom Crowther ist seit Oktober 2017 Assistenzprofessor für Global Ecosystem Ecology an der ETH und zwar im Institut für integrative Biologie. Tom beschreibt sein Labor auf seiner Webseite als, ich zitiere, "We're a collection of researchers, scientists and technical staff with a range of expertise from microbiology to digital marketing, all working with one thing in mind, the climate." Und damit passt Tom hervorragend in unser Departement, in dem sich sehr viele Professuren mit dem Klima und seinen Folgen auf Umweltsysteme beschäftigen. So with this topic, the climate, Tom fits perfectly in our department, where many professorships deal with climate and its impact on environmental systems. Tom Crowther wurde 1986 in Namibia geboren, hat dann in Cardiff seinen Bachelor of Science in Zoologie gemacht und promovierte 2012 in Ökologie ebenfalls in Cardiff. Danach verbrachte Tom seine Postdoc-Jahre an der Yale University in den USA und mit einem Marie Curie-Grant auch in Wageningen in Holland. Und obwohl Tom in seiner Wissenschaft klein angefangen hat, nämlich mit Bodenpilzen und Abbauraten und Bodenfauna, beschäftigt er sich heute mit Fragen zu ökologischen Prozessen, die Artengemeinschaften und Ökosysteme strukturieren, und zwar auf sehr unterschiedlichen Skalen. Und diese Skalen reichen von ganz klein, vom Holzstück, das im Wald abgebaut wird, bis hin zur globalen Skala, um zum Beispiel abzuschätzen, wie die Mikroorganismen im Waldboden die globale Kohlenstoffbilanz und damit auch den Klimawandel beeinflussen. Tom works across scales, from the very small ones like microorganisms to the very large one, the globe. He wants to understand how ecological processes affect communities and ecosystems and how this in turn affects, for example, the global carbon cycle. Bevor ich Tom persönlich kennenlernte, kannte ich einen seiner Fachartikel, der 2015 in Nature herauskam. Und es ging um die Anzahl von Bäumen auf dieser Welt. Es war eine Arbeit mit mehr als 30 Co-Autoren, beruhte auf mehr als 430.000 Inventarflächen auf allen Kontinenten, außer der Antarktis, keine Bäume da. Und es ging um die Baumverteilung in verschiedenen Biomen, vom Tropenwald bis hin zur Tundra, um die Faktoren, die diese Verteilung hervorruft und wie der Mensch die Anzahl der Bäume verändert hatte. Und diese Arbeit rief ein ganz erhebliches Medienecho hervor, von der New York Times zu National Geographic zur NZZ. Und bei der NZZ lautete der Titel "Mehr als vermutet, weniger als früher". Und Sie möchten nun wahrscheinlich wissen, wie viele Bäume hat es nun auf der Welt? Ungefähr für jeden Erdenbürger 422. Das heißt mehr als 3 Billionen Bäume und davon immerhin 1 Milliarde in der Schweiz. So before I got Tom personally, I knew one of his articles, which was even discussed in the local newspaper. This was the 2015 paper on tree numbers, and the end result was 3 trillion trees globally, about 1 billion in Switzerland. Doch damit nicht genug. Tom engagiert sich auch sehr stark, diese Anzahl der Bäume weiterhin ansteigen zu lassen. Die Organisation Plant for the Planet, ursprünglich eine Kinder- und Jugendinitiative, in der Zwischenzeit unterstützt von der UNEP und vielen nationalen und internationalen Organisationen, hat seit 2006 über 15,2 Milliarden Bäume gepflanzt. Tom fungiert jetzt als wissenschaftlicher Berater und vielleicht bald auch als Doktorvater des damals 9-jährigen Gründers dieser Initiative, The Plant for the Planet. Aber was Tom in der Zukunft vorhat, wird er uns gleich selbst berichten. Tom is also very actively involved in increasing the number of trees on that earth. For example, as scientific advisor for Plant for the Planet, and maybe soon as a doctoral father of the young founder. But what he plans to do here at USYS and at ETH, he will tell us himself. Tom, the floor is yours.
Thank you, Nina. You can hear me, yeah? Thank you, Nina. Thank you so much for the introduction. I didn't understand any of it, but I assume that the thanks are in order. And thank you very much for welcoming me here, everyone. And I'm very grateful for all of you for coming. I'm incredibly honored to be here representing ETH for the first time. And so I'm really grateful that we've got such a great turnout. So thank you very much. So I'd like to start just with a sort of course of events that have happened in the last couple of years that sort of contextualize some of the research that's that's motivating our research. A couple of years ago, The Independent covered one of my articles addressing how warming soils can stimulate the rate of climate change. And it was actually received very badly by several members of the Trump campaign, and there was a few furious tweets flying around. And actually, The Independent took this as an example, or used this study to exemplify several of Trump's questionable climate change policies in the last couple of years, and they've been repeatedly using this example over time. So this process actually somehow got me connected, sort of loosely connected to the first climate change lawsuit that happened in the United States, in the state of Washington, which led to the creation of the Clean Air Rule. And effectively, a group of lawyers They effectively sued the State Department on behalf of the organization called Plant for the Planet. And this is a youth organization that Nina mentioned earlier that are trying to restore the world's forests. And through this process, I actually became connected with this organization very strongly, and now I'm working with them, and they're soon to announce effectively the largest ever restoration campaign to try and restore the world's forests. And so ultimately, these are the kind of topics and societal impacts that are motivating our research, and this is what we hope to contribute to in the coming years. But for me, it actually all started a long time before this, in undergraduate university, where I was having a debate with one of my friends, who's actually a physicist, about the sort of relevance of life for our planet, for the functioning of our planet. And as an ecologist, I'm intrinsically interested. I believe that there's an intrinsic value of life, and that living things are different from non-living things. But his argument was that effectively living things are just blobs of molecules that adhere to the same physical and chemical processes that non-living organisms do, non-living things do. And obviously, biologists don't necessarily have a great definition for what life really is, and we certainly don't represent it particularly explicitly in Earth system models or in global scale models. So really it's a sort of philosophical question, but ultimately this is the kind of question that started me along this line, like trying to understand if the presence of life on our planet makes it function differently from any of the other planets. And I think one of the best examples of the relevance of life on Earth is the carbon cycle. This is a NASA simulation of carbon dioxide movement throughout the year. And you can see time ticking along on the bottom. And as you see, these are the high concentrations of carbon dioxide in the red up in the northern hemisphere. As we move into early summer, the emergence of trees on leaves in the northern hemisphere draws down that carbon dioxide from the atmosphere, and you can see the colors start to change. The red goes to yellow and then blue, and then all the carbon is ultimately taken out of the atmosphere. And so obviously this is an incredibly strong and relevant component of the carbon cycle that's driven by organisms, an ecological process. And this process strongly determines atmospheric carbon dioxide concentrations and the climate. And obviously a few months later, this carbon will be released back into the atmosphere through the decomposition of organic material and respiration of organisms. But it's these ecological processes that are driving the carbon cycle. And yet, one of the main limitations in a lot of these models is that the ecological processes aren't explicitly considered. And I think that's partially because ecology is a relatively new discipline. We're still interested in the fundamental questions at local scales, trying to understand how organisms are organized and interact with their environment, how the environment affects those organisms. But we're interested in our lab to try and turn it the other way. So we're interested to make sure that this fundamental research becomes relevant at a global scale so that we can understand the impact of those fundamental interactions for the global scale carbon cycle and the climate. And so when I talk about the organism's influence in the carbon cycle, this is ultimately the graph that's stimulating this research. So this is ultimately representing the sort of balance of carbon between the soil and the atmosphere, between the Earth's surface and the atmosphere. And these lines represent 11 predictions from 11 of the 11 Earth system models that we use to make our climate change predictions. The Intercontinental Panel on Climate Change uses these models to predict how the climate's going to change over the rest of the century. And as you can see, looking backwards from this time point, these models converge pretty well. They all predict that effectively uptake of carbon from the atmosphere onto the land surface and release of carbon into the atmosphere are relatively in balance, and so there's not much change over the previous century. But it's when we look into the future, you see this huge convergence of model predictions. And that's because we know humans are altering the carbon cycle, we know we're affecting ecosystems, and the different models have different interpretations of how the Earth system is going to respond, how this carbon balance is going to respond. Some models think we're going to get more carbon stored on the Earth. So a line going upwards indicates an uptake of carbon by the terrestrial ecosystems, and that would lead to less accelerated climate change. Whereas some of the other models are assuming that there's going to be losses of carbon from the soil into the atmosphere, driving the opposite thing, essentially accelerating the rate of climate change. And the lines that are going up simply assume that elevated CO2 in the atmosphere and global change drivers are going to stimulate the rate of photosynthesis, stimulate the growth of trees and the uptake of carbon into ecosystems. Whereas the lines that are going down assume that the stimulation is going to be on the side of the decomposition, that warming is going to stimulate the rate of decomposition and organic matter decomposition and the release of carbon into the atmosphere faster than the uptake. And so this would drive a very concerning accelerating carbon cycle feedback that could accelerate continuously into the future. And so much of my early research has actually addressed this topic, the impacts of warming on carbon, on microbial activity and carbon losses. And I did a range of studies looking at microbial communities and their interactions at fine spatial scales, looking at how ultimately changes in community composition and physiology of these organisms can stimulate the loss of carbon into the atmosphere. But during this process, I was realizing more and more that what I was doing is contributing to a huge literature base. There's probably thousands of studies on this topic, in some regions showing increases in carbon losses, others showing decreases under warming. And so it's really difficult to get a real global consensus on the magnitude of this feedback, and even identify whether it really exists. So I decided a couple of years ago to sort of go back to the drawing board and simply quantify changes in carbon stocks. And I did this by going to as many climate warming studies as I could around the world, and as many locations around the world as I could find where people have been warming the soil for a long time, warming the ecosystem, the plants and the soil for a long time. And I asked them if we could simply take soil carbon data from plots within these ecosystems. So we had a range of different ecosystem types, and they used a range of different warming strategies. But ultimately, in every single one of these locations, we simply had plots which were warmed adjacent to plots that were not warmed. And by measuring the carbon content in each of those plots, we can then calculate a difference for any site. Very simple. And then using some simple geographic information systems information, we can pair each of those locations up with the climate and the topography and the soil characteristics so that we can predict which types of ecosystems are likely to experience gains and which are likely to experience losses under warming. And what we were astonished to find out, actually, is that only one variable came out as a strong predictor of the change in carbon stock. And that was actually the amount of carbon in the soil at the beginning. So this x-axis here shows how much carbon there is in the soil in any region. And ultimately, in regions where there's not so much carbon in the soil, we see minimal changes in soil carbon stocks. This is the annual change in carbon stock per degree. So there's effectively no change in regions with little standing carbon stocks. But you see, as we move to the areas where there's large amounts of carbon being stored in the soil, these are the areas where you get large losses under warming. These are the places where you get big carbon losses. And because of the consistency of this line, and the fact that we know exactly, approximately how much carbon there is in every location across the world, we have global maps of soil carbon, we can then generate a spatially explicit understanding of this process. So we can predict which parts of the world experience losses and which experience gains. And simply, the pattern is the high latitude regions, where there's a huge buildup of standing carbon stocks over the last centuries of cold conditions. There's been limited decomposition. That means limited loss of carbon into the atmosphere. In these ecosystems, you get a huge buildup of soil carbon, and these are also the ecosystems which are likely to experience the greatest amounts of warming over the rest of the century. So it's in these high latitude regions where we're likely to get the big losses of soil carbon under warming, whereas the low latitude regions, the changes are negligible. And simply by adding up all of the pixels in each of these grids, the grid cells, we can get a global estimate of the change in carbon stock. So we can very simply estimate from the amount of carbon in the soil right now, how carbon stocks are going to go down over time. And actually, you should focus on the red line here, because this indicates the sort of business as usual scenario. The amount of, if we keep emitting carbon dioxide at the rate we do now, this is how stocks will continue to fall. It's approximately 1.5 petagrams, 1.5 gigatons of carbon per year. And that's about equivalent to having another industrialized country the size of the United States on the planet. So it's a really significant contribution. It's approximately 12 to 17% of current climate change. So it's really it has the potential to contribute to accelerating climate change. And because of the implications of this study, this is where the independent article came in. And we were all very excited. We all got very excited about the prospect of the media interest. And when we reached the front page of Yahoo News and Reddit News, and all my friends were texting me saying we're going up and up and up, and we were going towards the top spot. And we reached number 6, and then number 5, and then number 4. But ultimately, we realized that the top spot on both of those sites was taken by a picture of a dog. So we had no chance of making the top spot. But it put everything in context quite nicely. So I hate these dogs. But the relevance of the work becomes most apparent when we put it back into the Earth system models, which are taking into account lots of feedbacks like this. And so this is a different way of showing the Earth system model predictions by the end of the century. So each of these is one of the models, and essentially this dot indicates how much carbon is likely to change. They estimate that carbon would change by the end of the century. And a dot above the line is an increase in carbon stocks in the soil, and a dot below the line is a loss of carbon stocks into the atmosphere. And so you can see there's quite a big spread. But when we when we put our spatially explicit information into these models, we can see two things. First, the dots come down. So we're having a greater likelihood of losses of carbon into the atmosphere. But more importantly, almost, the uncertainty is almost halved. So simply by generating a global understanding of this ecosystem process, we can minimize some of the uncertainty in this critical, it's a very important global scale process determining the climate. The other possible downside is now that we actually have replicable data points, we've got variability within each of our estimates. And all of these model outputs are just the average model output. But sadly, now when we have our full data, we actually have parameter uncertainty, and actually the variability within any single model is actually greater than the variability across the models. And so this parameter uncertainty is something that we really want to pin down in the second half of my talk, and I'm going to be talking about that now. So anyway, I've just given a sort of summary as to one of the overarching questions in our lab. We're interested in how warming alters the carbon balance in each of the pixels around the world, so we can get a global perspective. And we're looking at the moment we've currently got a study on the impact of nutrient enrichment on the global carbon stocks, and we're starting studies on drought and CO2 fertilization as well to see how all of these global change factors are going to change the balance of carbon in terrestrial ecosystems. But the one limitation I see at the moment in our ability to make fine-scale predictions is that we make these, essentially we run equations at each of these pixels, and within each pixel we know all the sorts of information about the abiotic characteristics, so that is the environmental characteristics. We know the climate, we know the soil characteristics, the soil chemistry and physics, we know plenty of things about the non-living components. But even though it's the organisms that are driving the carbon cycle, we know very, very little about the organisms that are driving the carbon cycle in those pixels. So if you had two identical red pixels next to one another, one could be a forest and one could be a grassland, but they're effectively being treated exactly the same in these scale models. So the second half of the sort of second goal in our research is to generate fundamental understanding of the functional biogeography of the important organisms that drive the carbon cycle. And we're not necessarily interested in where individual species live, but we want to know what the organisms are doing in each of those pixels. So as we start the lab, we're a couple of months in now, we've started projects on both soil invertebrates and bacteria around the world. But I'm going to talk about the ones that I've been focusing on until now, which are strong drivers of the global carbon cycle, the trees and fungi. So I'm going to start by talking about the functional biogeography of fungi. And so I'm going to keep this panel along the side just so you can remember that we're talking about fungi here, and this is the section of the talk that I'm on. Actually, I'll grab this. I forgot my bottle. Thanks. So when we think about fungi, I think most of us think about toadstools and mushrooms that we see in the forest. And these are obviously a very functionally important part of the fungus, involved in the sort of reproductory process of the fungus. But actually, the main component of the fungus, the most functionally important part of the fungus, is occurring below the surface. This huge mycelial network of mycelial cords that interconnect forest trees, and they go throughout grasslands and natural ecosystems. And these fungi, these hyphae, are responsible for the distribution of nutrients throughout their ecosystems, and they're ultimately, most importantly, responsible for the degradation of organic material. They convert dead organic material into nutrients that can be taken up by other organisms, but through this process, they determine how much of the carbon is released into the atmosphere and how much is ultimately stored in the ecosystem. These fungi physically give soil its structure. They bind aggregates of soil together, and they form microhabitats that other organisms can live in. They keep the soil a living thing, ultimately. And even the decomposition of that fungal necromass, when the fungi die, the decomposition of that necromass is the process through which soil organic matter is actually formed. So these fungi really determine the balance of carbon between the soil and the atmosphere. And even though we know different fungal groups will do these things at completely different rates and in completely different ways. One fungus will decompose wood 100 times faster than another fungus, and yet we know nothing about which individuals live where. And that's primarily, it's probably driven by the hyper-complex nature of fungal communities, which has sort of forced us down the line of having to use genetic sequencing approaches to characterize their global distributions. So the majority of global-scale fungal ecology studies have used genetic sequencing approaches to try and characterize all the thousands of species that live in each patch around the world. And what they consistently find is that the further away you get, the more dissimilar the fungal communities get. So the further away you are, the more different combinations of species you get. And this has led some people to assume that fungi are somehow fundamentally unique. They're different from all the other organisms in their biogeography because there are no meaningful patterns in their biogeography. You don't see clustering of some species in some regions and clustering of other species in other regions. Now, like fine-scale microbiologists that are focused on individual regions will say that there's plenty of evidence for that, but the global patterns never seem to provide any evidence for clustering of fungal groups. But I actually think if we applied the approaches that we use to study fungi to plants, we would see a very similar thing. I've got here just an image, a basic image of the general biomes that we see for plants, that is, they're indicating different plant types. And we can all of us know the difference between a desert plant and a forest plant, per se. But if we looked at these plants the same way we study fungi, if we just generically sequenced them and identified their species composition, the species that are existing in the desert in Australia are taxonomically very distinct from the species that are existing in the desert in North America. It's only when we look at their functional attributes, their traits, that we really see the patterns. If we simply looked at the species composition, it would be much more difficult to see the meaningful patterns. But when we look at the characteristics of the organisms that allow them to live in those ecosystems, we can start to see broad biogeographic patterns. Again, all of us can tell what a grassland plant looks like next to a forest plant. We all know what a tree is compared to a cactus. And these are the kind of basic bits of information we want to try and generate by using a trait-based approach to understand fungi. And so what we did is we collected fungi we were sent fungi from all over North America. And they were a range of different species from a range of different phylogenetic groups, but ultimately they were all functionally equivalent wood decomposition fungi. So they govern the rate-limiting steps in the decomposition of wood. And they release carbon from decaying organic material, and they release it into the atmosphere and also sequester some of it in the soil. And so we have the full phylogenetic history of these individuals, but they were a random subsample of fungi from across North America. These are very common, ubiquitous species, and they grow in a lot of different places. And if they were growing in the same place, they would be competing with one another to sort of colonize wood. But ultimately, they were all collected from different places. And what we wanted to do is simply look at their hyphae, look at those fungal networks, to see if we can see any characteristics of those hyphae that can help us to discern which ones are from the dry regions and which ones are from the wet regions, which ones are from the Arctic and which ones are from the tropics, because we had a huge climatic variation. So we actually measured a wide range of functional traits on these fungi. We measured their growth rate, their morphology, their chemical composition. We measured their enzyme production and their volatile organic compound production and their cell structures, all sorts of things that might help us to discern where they come from. And the most important, I'm going to focus on some of the most important traits that we measured during this process. And actually, this is one that we thought would be our best predictor of where the fungi come from. So we measured, this is just a physiological growth response curve. So we grow each of our fungi along a range of different temperatures, and then we simply measure how quickly they grow at each of those temperatures. And we thought, okay, this is going to be great. We'll grow them at all the temperatures. The fungi from the tropics will grow optimally under 30 degrees, and the fungi from the Arctic will grow optimally over here, and the point at which they grow best will tell us where they come from. There's a trait that relates to the environment. And so we grew all of our fungi across a range of environments, and we found our initial theory was entirely wrong. Everyone wants to live in the tropics. All the fungi that we had, whether they're from the Arctic or the tropics, they all wanted to live between 25 and 35 degrees. So we thought, okay, either we've failed or they don't use temperature as a selective, maybe it's not a very good selective pressure. Maybe they're living under the soil and they don't really care about temperature. So we thought we'd do it over a gradient of moisture, because that's a much stronger selection pressure, and we found exactly the same thing. So all of our fungi they grew most optimally under moist and warm conditions. Everyone wants to live in the tropics. But as we explored the data set further, we realized the most meaningful axis of variation isn't the highest, the maximum place where they grow, the fastest growth rate. It's actually their niche width. So a lot of the fungi had very narrow niche widths, so that's a very narrow hump, and they can't survive under very dry conditions, whereas others can grow under incredibly dry conditions. And we think this variation maybe is something meaningful. But it makes us think the individuals that are growing here can grow everywhere, whereas these individuals simply couldn't exist here. So for these ones to exist, there must necessarily be some sort of trade-off that allows these ones to be incredibly competitive under the optimal conditions. Otherwise, they just wouldn't exist anywhere. And so this is a concept of a trade-off, a dominance-tolerance trade-off, which is certainly not a new concept in community ecology. It's a very old concept. The idea is simply that your ability to do something or to express a trait necessarily comes at the expense of your ability to do something else. And so we sort of proposed based on this that the individuals that are able to live in the tropics are the ones that are able to fight, while the individuals that are living in the desert are simply able to tolerate stress. And the ability to tolerate stress comes at the expense of your ability to fight. But the only way for us to find this out was to fight all of our fungi against one another. So this was about 1,600 fungal combinations. It took a hell of a lot of time, and a lot of people lined up doing Petri dishes in our lab. But ultimately, it was a fun process because they made loads of pretty colors and did loads of interesting things. And we measured the growth rate towards one another and the growth rate over one another and under one another. And we ultimately used a range of different approaches to characterize which individuals had won. And this allows us to come up with a ranking. And finally, I was delighted to find that one of our theories was correct, that actually we do find evidence for this dominance-tolerance trade-off. So that's simply saying the individuals that are highly competitive are very bad at tolerating climate stress, whereas the individuals that are stress tolerant, they are very non-competitive under optimal conditions. So this certainly isn't the first time anyone's shown a dominance-tolerance trade-off in a group of organisms, but it is one of the first times that it's been shown at such a broad spatial scale, and certainly with a group of fungi. And the reason that this is possibly the most exciting graph that I've generated during my research is because I find it fascinating that we've got a fungus that's growing in a log up in Alaska, and another fungus growing in another log in the tropics, and they're ultimately governed, their distribution, their existence where they are, is governed by this fundamental relationship, this fundamental trade-off that is acting across both of them. They're both either, the one in the Arctic is unable to be competitive because of its ability to tolerate harsh conditions, and the one in the tropics is highly competitive despite its ability to not tolerate harsh conditions. Sorry. And so when we throw all of this information into a principal component analysis, this is a different kind of graph that essentially it allows us to explore the differences between all of our fungi. And if we throw all of our traits in, I realize it's a bit of a complicated graph, but we're just trying to see which sort of traits correlate with the differences in our fungi. When we include all of our 157 measured traits, this dominance-tolerance trade-off comes out as the most important axis of variation. So the individuals on the right are the fast-growing, they grow optimally under optimal conditions, they are highly competitive, whereas the individuals to the left are stress-tolerant. They can survive under cold and dry conditions. And this dominance-tolerance trade-off correlated perfectly with a well-known trade-off in growth ability. So if you imagine as a fungal hyphae, you grow by producing tubes. And so you can either grow your tubes very long and fast into one direction, or you can grow them all on top of one another. And the individuals over here are investing all of their growth on top of one another. So they're growing sort of thick, dense mats, where the individuals over here are growing fine tubes. And this growth difference seems to be underpinning everything that we're seeing. So the fungi to the left is a sort of exemplified by this fungus here. It's very thick and dense, and it's got a buildup of hyphae that's protecting it from the harsh environment. But that by allocating all of its growth on top of itself, it can't extend very rapidly. Whereas the individual on the right is growing incredibly rapidly, and it's growing over its opponent and destroying its stress-tolerant competitor. So these simple traits seem to be explaining some patterns underpinning these organisms. But the relevance of this comes when we link it back to the environments that they were collected from. If you remember, we've got fungi in the Arctic and fungi in the tropics, and we're trying to tell which ones go where. And if we look at any of the individual traits, any of the single traits on their own that we've measured, for example, here, the climate stress tolerance would be the temperature niche width. They don't correlate with any of our environmental characteristics. We know all sorts of things about the environments that they were collected from. We know the temperature, the moisture, the soil characteristics, all of these things. We can't predict anything based on individual traits. But when we convert that trait to a dominance-tolerance trade-off, so simply divide the competitive ability by the climate stress tolerance, finally it sort of stretches out our axes and we start to make meaningful sense. We start to see method in the madness. There's a pattern here. We can see in the warm climates, these are the competitive ones relative to their tolerance ability. And it wasn't just temperature that correlates with this trait, this new trait. We also had the aridity of the soil, the amount of moisture available in the soil, the texture of the soil, the pH of the soil, and the organic matter content all work together to sort of structure this dominance tolerance trait. And because we know all of this information at every single pixel across North America, we can generate a spatially explicit map of this dominance tolerance trait. This physiological trait, we can finally say some characteristics of these organisms that allow them to exist in one place rather than another. And we can finally say the individuals in the red parts are more competitive and the individuals in the white parts are more stress tolerant. So this basic functional biogeographic information is useful. It's very interesting for us to understand where fungi exist, but it's most valuable when we link this trade-off, this trait, to the actual functional attributes, the attributes that tell us how they are affecting the carbon cycle. So we've got a postdoc in our lab, Nikki Lustenhauer, at the moment, who's currently exploring that, looking at how this dominance tolerance trait relates to the wood decomposition by each of those fungi. So we've grown all of our fungi on little blocks of wood, and we simply measure how quickly those bits of wood lose mass. And that's the decomposition rate. And we find very strongly that the individuals with a high dominance tolerance, the competitive, fast-growing ones, the thin tubes, those are the individuals that are decomposing very rapidly. They decompose that wood fast. Whereas the individuals that are growing slowly and stress-tolerant individuals, they don't invest much in decomposition, so they're growing very much more slowly. And so because of this simple linear relationship, our functional biogeography map can turn into a map of intrinsic fungal decomposition rate. We can say the fungi here are fast decomposers and the fungi here are slow decomposers. And even more interestingly to us is that the fungal structure, the hyphal structure, is even different between these groups. The individuals up in harsh, cold, difficult conditions, they invest a lot in these calcium oxalate crystals. So there's these jagged little crystals across all of the hyphae that are they're sort of protecting the hyphae from the outside world, and they're also making it a very harsh, recalcitrant structure. Whereas the individuals from the tropics are all fine, beautiful, thin tubes with no calcium oxalate along them. And they actually have a low carbon to nitrogen ratio, so they're very easily decomposed. So when these fungi die, these ones are incorporated much more efficiently into the soil carbon, whereas these ones are decomposed very inefficiently, and a lot of the carbon is released back into the atmosphere. So the structure of this dominance tolerance trait is starting to tell us not only how these fungi govern carbon loss from the ecosystem, but also the carbon stabilization within the soil. So this is obviously the very first steps in our exploration of these traits, and of course we've only used 45 fungi. And we need to explore this using a range of 1,000 fungi to confirm or to test these patterns. But the next steps that we're going to look into are how the sort of interactions between multiple species allow us to understand the sort of maintenance of diversity within each one of these pixels, so that we can try and look at the trait dispersion, so we can understand how variable these traits are within every pixel. At the moment, this is a map of just the average for each pixel. But this is just the very beginning. I'm just giving you a glance. So the next thing I'm going to talk about is the trees. And Nina mentioned the study we did earlier. I'm going to be talking about that right now. So we're interested in, ultimately, I don't need to persuade anyone how valuable trees are for the global carbon cycle, but trying to understand the global distribution of these forests. And again, I'll keep this panel along the left-hand side so you can remember I'm now talking about the biogeography of trees. So when it comes to the trees, we have a completely different set of problems. Fungi are all hidden below the surface, and they're very complex. We can see forests from space, so it's a different set of tools, a different set of questions that we're addressing here. We can see, obviously, there's a rich history of forest ecology that tells us, and forestry that tells us all sorts of information about local forests and how to manage them and how they're structured and how they interact. But our understanding of global scale, the global forest system, ultimately comes from satellite information, which tells us where forests are, and a little bit of information about how they can exchange carbon. But they don't tell us that much about what's going on below the surface. And this is the part where Plant for the Planet came into my life. So I became involved with this organization that were restoring trees around the world, and they were actually leading the UN's billion tree campaigns. They planned to plant 1 billion trees around the world to help our climate problems. The problem was we didn't know how many trees there were to start with, so they couldn't really generate an understanding of the magnitude of their impact. They couldn't tell quite the extent of their efforts and really set meaningful restoration targets. Oh, sugar. So this is where I got involved, and I wasn't necessarily interested in the global tree number per se. I'm not so focused on population numbers, but I was interested in the idea because in order to do that, we needed to generate a spatial understanding of forest structure. And this is, well, I've got a video that will talk you through the process.
Trees are some of the most prominent organisms on the planet. They prevent soil erosion, store water and nutrients, provide habitats for thousands of plants and animals, help offset the damaging impacts of climate change by absorbing CO2, produce oxygen that we need to breathe, and directly improve human happiness and well-being. Because of all these benefits, the United Nations Environment Program and various other organizations have invested huge amounts of time and money in order to plant 1 billion trees around the world. However, the impact of this effort is uncertain, because until recently, no one knew how many trees there were to begin with. Would planting 1 billion trees increase the total count by 1% or 50%? Could 1 billion trees help our climate problems, or would it just be a drop in the bucket? Our current understanding of the global forest extent comes from satellite images, which provide valuable information about forest area and canopy cover, but they cannot tell us about everything that is going on below the surface. So, a research team from Yale University in the School of Forestry and Environmental Studies set out to map the world's trees. With help from scientists all over the globe, they collected thousands of ground-sourced tree density measurements in hectare-sized plots. They paired tree density in each plot with satellite images of forest cover. and they integrated information about soil type, topography, climate, and human disturbance. Using all of this information, they constructed predictive equations which allowed them to estimate how many trees are likely to exist in any given area, based on its similarity to environments with known tree numbers. The resulting map is the first of its kind. It reveals not only where all the trees on Earth are located, but it also tells us about the density and environmental structure of the world's forests. It can help us to understand which environmental conditions are best for supporting trees, which habitats might be suitable for plants and animals around the world, and how carbon storage varies across landscapes. By adding up the values in each square kilometer pixel, the Yale team was able to estimate that the total number of trees on Earth stands at about 3 trillion. This number was considerably higher than scientists had previously expected. The study also shows that we have reduced the number of trees by almost half since the start of human civilization, and we're losing 15 billion trees each year. In other words, the number of trees cut down every year is twice as big as the total number of humans on Earth.
So now we all have the answer to a nice pub quiz question. Ultimately, the sort of end result was for Plant for the Planet. We showed that by using a combination of ground source data and satellite information, we estimated there's approximately 3 trillion trees on Earth. And obviously, the relevance of this for them was very useful. If you imagine each of these trees here is 1 billion trees, this is how many trees currently exist on Earth. And this was the contribution of the Billion Tree Campaign, which is great, but it's not the magnitude of the impact that they're looking for. So it's now the Trillion Tree Campaign, and when they achieve this, they're really going to have a meaningful impact at a global scale. And obviously, this sounds like a ludicrous goal, but since that study was published, they've already planted 14 billion new trees. So they've really, having these new targets, setting these new strategies has allowed them to really ramp up their efforts, and they're moving incredibly well towards their goals, and maybe one day they'll reach it. And this spatially explicit map of tree populations helps us also to understand how many trees are lost each year and the potential for new forest gain if we were to restore around the world. So there's possible space for another 1.3 trillion trees. But the value for me of this global forest map isn't just in the tree numbers. Ultimately, we've made a spatially explicit map of tree density. And for anyone who's walked around in some forests, you'll know the difference between an open forest and a closed forest, a very dense forest. Open forests will have a very different subset of understory vegetation. Different plants and animals can exist there. Even indirectly, carbon and nutrients and water are recycled differently within these ecosystems. So the density of the forest is actually a really important habitat or component of the habitat structure. So these maps have now been useful in understanding and modeling species distributions and biodiversity patterns at a global scale, because they can help us to say which species might be able to live in which regions. And actually, it was recently used in that map that I showed you at the very beginning as one of the very simple base layers of information to provide a small amount of ecological information that can be helped to parameterize those global scale carbon cycle models. But obviously, the density of trees is a useful indirect piece of information about the forest structure. But what we're ultimately interested in is the carbon storage and the carbon cycle. So we've now stepped up our efforts. That original study was based on 400,000 plots of tree density. And we've now got 1.2 million forest inventory plots that include information about stem density, but also the basal area. So that's the sort of the size of the trees within each of those plots, and also the species identity of every single tree in those plots. So this is a huge spatial coverage of the world's forests, where we have real on-the-ground information calculated by foresters standing on the ground. And by simply pairing that information up with the globally available satellite information, we can really improve our picture of the global forest system. So the first thing we did actually was to explore a very simple thing, but an ongoing question in community ecology, and that's the relationship between the number of species in a plot and the amount of productivity, the amount of biomass that's produced by that plot. And we find consistently at a global scale, plots with more species have a greater potential to capture carbon and to put on biomass and to fill their space. And the idea behind that is a simple one. Ultimately, communities comprised of the same species will be competing in the same way for the same resources, and they'll clash with one another. They're not able to capture the light and nutrients as efficiently as they might hope. Whereas plots comprised of very different species have different strategies for capturing light, different strategies for capturing nutrients and water, and so they'll fill different niches, and they'll be able to more efficiently capture all of those resources and grow more effectively. And the consequence of that study actually there was quite clear economic implications. Because in terms of timber, the value of the world's forests at the moment are estimated at for the timber industry and pulp and paper globally, are estimated at approximately $616 billion per year. And there's ongoing debate in different regions of the world whether it's worth replacing those forests with monocultures of the most productive forest, the most productive tree species, because maybe that will be capturing a lot more carbon and producing a lot more timber. But our estimate was if we were to do that globally, convert the world's forest into a monoculture of the most productive species, the annual budget in terms of timber would fall to about $406 billion. So that's a $200 billion deficit each year. And that's actually 7 or 8 times greater than the total amount of money that we currently spend on forest conservation. So it's sort of an economic reason to be conserving diversity within our forests at a global scale. And actually, this is very preliminary information, so I should stress this is the conclusion that I stressed earlier about the relationship between species diversity and productivity. That's a global scale conclusion. So we're not looking at the regions in which these relationships matter. So we're starting to break that down now, and this is some very preliminary evidence that's been compiled by a master's student in our lab, Haozhe Ma, and he's actually looking at the relationships between species within each of the pixels around the world. So he's combined various pixels, and he's looking at whether species do facilitate one another or if they actually antagonize one another and actually compete more aggressively against one another. And he's finding evidence that in the warm and moist parts of the world, we're actually seeing that individuals are competing for those optimal resources, those optimal conditions. They're competing incredibly aggressively, and actually there's negative associations between species, whereas it's the positive facilitative interactions we're seeing much more commonly in the dry or cold parts of the world. Where it's harsh, you then start to see facilitation a lot more. So it's not necessarily a simple global relationship. We need to start to understand these patterns and understand the ways in which plants are interacting with one another before we can say where diversity is most valuable for productivity. But of course, the main thing we're going to be doing with these global data sets is simply generating spatially explicit information about the global forest system. So we're currently involved in projects looking at biomass production, productivity, basic attributes of the global forest system that we are very poorly constrained at the moment, very, very poorly understood at the moment. And this basic information is going to be very valuable for our ability to predict the carbon cycle. And we're also looking at various traits that might be distributed across the world's forests so we can understand the structure of these forests very well. But I'm just showing you here a map that we've recently finished and we've just submitted. This is a map of the mycorrhizal status of the world's forests. Now that we know information about all the types of species of tree that exist across the world, every tree is affiliated with a certain group of mycorrhizal fungi. And because now we know where all the trees are, we can say something about the types of fungi that are existing below the soil. So actually, by having this global scale above ground information, we're starting to get new insights into the way that the below ground world is structured. And this will be one of the first global soil biodiversity type maps. So I hope in my talk I've sort of explained some of the concepts that we're addressing in our lab. We're very much interested in generating a fundamental, basic understanding of the fine-scale processes that structure communities. And we're not just looking at the broad-scale implications. We focus on the mechanisms that are getting there. But we are very much focused on extrapolating that information up so that we can make real global implications that can have impacts for our understanding and our ability to address climate change. And obviously, our approach in doing this, generating this holistic view of the world's forests, is an incredibly interdisciplinary type of thing. We use understanding of Earth system dynamics to better understand how all the different global change drivers will explain the balance of carbon in every pixel around the world. And we'd like to say things like, understand how the addition of forest to a specific pixel might warm or cool the global atmosphere. But in order to do that kind of research, we're very much focused, we're generating spatially explicit information about the functional biogeography of these important organisms that govern the carbon cycle. And these are very different skill sets than the first. And ultimately, it's all underpinned by very basic community ecology, understanding how diversity is established and maintained and distributed across broad spatial scales. So these combination of approaches provides us with this holistic view. But it's a very different set of approaches. These people are using remote sensing skills and geospatial analyses, whereas the soil biodiversity people are interested in genetic sequencing approaches and culturing. And of course, there's basic community ecology theory and earth system modeling theory that are all very different disciplines. And this was my concern as we started developing this lab. How was I going to sort of have this interdisciplinarity within our lab? But we've recently partnered with an organization called D-USYS, and this is the foundation that sort of shares our vision for this interdisciplinary nature of global scale research. And they're very sort of excited by the prospect of having this interdisciplinary type of lab. And so with their sort of support, we're now establishing sort of 4 groups within our lab. So 2 senior postdocs associated with each of those 4 disciplines, experts in those regions. So it's not necessarily a very top-down lab system where it's a professor with experts in exactly the same topics. We've got a range of experts from all of these different topics communicating with one another and sharing their information so they can get a more holistic understanding of the system. And we're also in the process of recruiting some PhD students associated with each of those groups as well, PhD students and master students who are working on these kind of topics. So we're in the process of building at the moment. And of course, we've got there's a strong emphasis on our support structure because having such an interdisciplinary group means that we need to have a good sort of good group behind the scenes. And so we've got fantastic administrative support and technical support with programmers and lab technicians. And one of the people that DOB Ecology really encouraged us to include within the group is a strategy and development officer. This is someone who can understand the environmental landscape and understand how policies and systems are changing within the environmental movement so that we can stay on top of things, so we can stay relevant rather than getting focused on particular niches or going down rabbit holes. But the most important thing that they stress to us is that they only found out about me and our research because of the media impact and the communications, the social communications. So they've stressed the absolute necessity for us to invest real time in digital marketing and communications so that we can make sure that our messages are being consistently communicated to the public and not just sort of beyond the scientific sphere, making sure that we get our messages out to the real world. And actually, the communications team has come up with a short video that's going to summarize some of the fun activities that have been happening in our lab in the first couple of months.